And our last speaker for today is Dr. Rachael Sheridan, the Flow Cytometry Core Manager at the Van Andel Institute. And her talk title is Approaches for Handling Challenging Samples, something that those of us who run a core are faced with on a fairly regular basis. So thank you, Rachael.
Right, thank you so much for inviting me. Now I'm going to fight PowerPoint to get this started. Great. Can you guys see my slides?
You're all good, Rachael.
Oh, good. Thank you. So my talk's going to be a little less scientific and a little bit more appropriate for a year like 2020, I guess, and just some ways that you can approach things when things haven't gone quite as planned, specifically from the biomedical standpoint. So these salvage operations are just a part of life. We design the perfect panels in theory, and we give users the protocols that we've seen work before, and then they come to the lab and we start looking at it, and this is what we end up with. Now, it may not be as pretty, but it's still cake. So sometimes we just have to see what we can still get out of the data that will help us to improve or entirely salvage an experiment if possible. So unfortunately, I don't get a whole lot of cake at work, but I do get a broad range of samples into the lab. Because my core here at the Van Andel Institute, it's in Grand Rapids, Michigan. I know nobody's heard of it. We're working on that. We have a very much biomedical focus. Okay, so I will get samples ranging from the easy stuff like blood. I really like working with blood. It was made for flow cytometers. Okay, that's how this works. And I think a nice phycol prep is just one of the happiest things to see the perfect layers pop up. We deal with bone marrow from mice, from humans, from rats. It's also pretty easy. You know, it's already a single cell suspension. Again, pretty much made for a flow cytometer. Just filter it because cytometers don't like bone shards. Then we have, you know, your typical cell cultures, adherent suspension, lymphoid tissues, and other tissues. And some of these can get pretty gross. So we got things like brain, intestines, mammary epithelium, solid tumors, fallopian tubes. We've seen all sorts of things. And some of these get very difficult. So sample prep is so important to having a successful flow cytometry experiment. Now, when you're working with cell lines, there are some things that you can think about. Usually, well, if you have a suspension cell line, cool. I mean, pipette them out, spin them down. But if you have an adherent cell line, you have some choices to make. There are various methods of mechanical, chemical, and enzymatic digestion. Some people like to scrape their cells, but I would really discourage this. Every time I see it, it looks terrible. It often will cause a good portion of your cells to disrupt their membrane, and then they'll show up dead, or you end up with bare nuclei, also dead. Okay. So I really would like to discourage that. And you may have to try different enzymes to see what works best. Trypsin, EEE, what's the other one I used to use? I'm blanking on it now. Accutase, that's it. Okay. And how you quench these enzymes to prevent overdigestion can also impact your sample quality. Some cells, it doesn't really matter. You can use media or your FBS to quench, and that's fine. But some cells, they will pick up on everything in there, and it makes them sticky again. So if you are stripping with trypsin, you could try using soybean trypsin inhibitor. We've found that in some of the cell lines that are fussier and tend to reaggregate, that the soybean trypsin inhibitor quench reduces some of that aggregation. A large portion of cell-cell aggregation is due to the proteins that require divalent cations. So try to have buffers without calcium and magnesium if you can. We also usually include like 2 millimolar EDTA. For cell lines, again, if you're gonna be sorting them, you may want to include some Hebes, just because this can help with viability during sorts. These cell lines aren't too bad. Then we get into tissues. And unfortunately, I don't have any magical recipes for tissues. Every tissue is different. Every tissue, depending on how long it's been out, how you've stored it. Most of these are going to take some combination of mechanical and enzymatic digestion, but you will need to figure that out. So determine this empirically, try different enzyme types, ratios, time, temperature. And when you're planning these experiments, think about your target cell population. Is this a cell population that's located closer to the surface of your tissue so it'll digest out faster? Or is it deeper in and you're going to have to get farther into the tissue and make a longer digestion time? How are you going to know when you've got a good digestion setup? So you need to have these metrics to judge these dissociation outcomes during your protocol development. Do you need a specific surface marker to survive? Do you care more about viability? Quick and dirty flow panels are pretty good for judging this. The one thing that I do offer for any users who use our tissue dissociator is that they can do viability on like the Cytoflex or something, and we'll help them do that really fast. Yeah. So in addition to published literature, there are some other resources. If you have no idea where to start, Worthington Tissue Dissociation Guide has a group of publications that's searchable by tissue type and species. So it can get you down the right road and you can see, okay, well, how are people digesting mouse lungs or, I don't know, other, whatever piece you want to digest, intestines, I don't know. And then some of the companies selling enzymes have various guides for different tissues too. Now, one of the things about doing these sort of tissue dissociations is once you have a protocol, great. But as part of this, track your reagents. So just a quick case study from my lab. I had a user, she was dissociating a tissue and had been sorting for GFP positive cells. It usually was about 5% of the total and it went really well. But one day, it dropped to less than 1%. And we thought it was a fluke. She tried again. It was really, really low. Then they tried to blame the sorter, but the sorter QC'd just fine. Everything was okay. Note she didn't have a viability dye in this panel and had no way of really QCing her sample. So after a lot of back and forth, we convinced her to do it one more time and just take a part of her sample and stain it with DAPI so we could see what was going on. And this is what we saw. It was 80% dead. So her target cell population was mostly dead. Right? And therefore, she wasn't getting what she needed out of her sorts. So what she did is she went back and we were like, what has changed? And she had a new bottle of trypsin. She dropped the digestion time by 15 minutes and ended up with 90+% viable cells. So now, because she still fears putting the viability dye in her sort sample, we've compromised. And she takes an aliquot of each sort sample and we do a pre-sort viability check on that before proceeding. And now her GFP positives are back up into that 5 to 8% range, right where they need to be. Right? So just because you have an optimized protocol doesn't mean it's going to work forever because reagent lots change. And this can go for antibodies and fluorochromes too. Just because you've titrated one, if you change your supplier or you get a new lot, it may behave differently. So think about that in your polychromatic flow panels and having a validation step for your antibody before you just keep going with it. There. So in addition to making sure you get good viable cell preps, you also need to think a bit about how to stabilize these single cell suspensions. Filtering, always a good idea. If you think you've got like smaller aggregates and you look at it and it's kind of like cloudy or maybe has some dandruff, you might be looking at some cell-cell adhesion proteins. So again, EDTA. If, however, you look at it and it looks kind of like this snow globe here, and you see stringy stuff in it, that's a sign that you have really high levels of cell death and that you should incorporate a DNase treatment. So typically like 25 to 50, I think that's supposed to be micrograms per mil, I'm sorry, of DNase 1. And DNase does require magnesium to be functional. So if you're going to do a DNase treatment, no EDTA around, do add some magnesium. And you can treat them with a higher concentration of DNase first, like during processing or during tissue dissociation, and then drop it to a lower one to maintain the solution after you've got them all prepped and stained and headed to the flow core. Another thing to think about is autofluorescence. And this one's a little weird. It's kind of a niche case, but autofluorescence, although we often think of it as being annoying and something we have to work around, it's occasionally useful. This is a case of where this particular example is where it's a little bit annoying. These were nuclei isolated from mouse brains that weren't very well purified, so we had so much myelin in there. And when my user brought me the same sample split, and she stained one half of the nuclei with DAPI and one half with PI, we ended up seeing that we got much better signal from the PI because all the junk in there autofluoresced where we were looking for DAPI, but not so much off that yellow-green laser. I also use autofluorescence in some of my other panels to help distinguish macrophages from other cells. It's like, oh, yep, those are more autofluorescent than these lineage positive ones, so like my lymphocytes. And it kind of gives me a nice sanity check within the data to make sure that I'm identifying the right cell populations. Now, when I really have to salvage something or I'm just not sure what's going on, what I often will do is just mix it up, rebel against that standard order of gate on scatter, gate on singlet, gate on live. If there's a lot of cell death and debris, I often will go for a live gate first. Okay, so these ones, I could probably gate that pretty well on scatter. This one, it's like, I don't know. So if I switched it and I looked at maybe forward scatter versus my viability dye here, then I get this really nice separation of where my live cells are, and I get rid of a lot of dead stuff, debris, and noise all at the same time. It's especially helpful here when I have this mouse intestinal prep. These preps are not pretty. Okay, so now we can see this population really well for our live cells. And there's nothing that says you have to start with viability either. Some other ones where I'm desperately trying to find a moderately rare cell type amongst a field of dead and debris, I'll actually look for the markers I want first and then go backwards and be like, okay, so the things I want are probably in here. Now let's see where they fall in viability and scatter and everything else. So try a few different ways. You never know what's going to help you out the most. Another trick that I've found is very helpful is to use more than one dye. So you can combine cell permeable and cell impermeable dyes to identify your viable cells from debris and dying cells. So this is probably my favorite example. I wish I had taken a real screenshot when I did this one. So we had a user who was bringing us maize pollen granules that he dissociated and was trying to ground up. I don't even know how he does that. And then to get the sperm cells out of them. And we kept sorting for what we thought was it, and it wasn't, and it wasn't. So we added in a second dye. So we've got propydium iodide and Hoechst for this one. So the propydium iodide is going to go in and stain everything that's dead and has nucleic DNA. The Hoechst was going to get into even live cells and stain it. So this showed us, the Hoechst was going to show us all of the live stuff. And then the PI was going to say all of the stuff that has a nucleus. And the PI was going to tell us whether it was live or dead. And this waterfall pattern was just really cool. And this works great in mammalian cells too. So here's one of those intestinal preps again. And this time we've used dicycle ruby and DAPI. Okay, so here's what it looked like when we looked at just the DAPI, but now I add in dicycle ruby. And here are my cells. And they are DAPI negative, dicycle ruby positive, so these are the live ones. So if you try to use just one of these on their own, like dicycle ruby, please do remember that it will stain the dead cells too. It will stain anything with a nucleus. You can see these guys are somewhat dicycle ruby positive. But what does happen is when you then add in the one that stains just the dead cells, if you add it at enough excess, you can see it start to kick the other one off because some of them compete for binding. So like propydium iodide is an intercalator, and it was messing up the minor groove that the hooks required. So they kind of did this. It looks like there's probably less competition for binding with these two, although I didn't throw in quite the excess of DAPI that I did for PI in that other experiment. So using the two of these dyes together can be really helpful. Or sometimes, maybe your user brings you a panel where they've got PE and PI together. Can you gate it? Yes. But there was also no controls and a very low number of cells. So as a sanity check before we did anything, here's what it looked like with the PE versus the PI. And these are what I would have guessed were my dead cells, and these probably would have been positive, but wanted to check. Here's looking a little bit farther out, hoping to catch more PI than PE, which kind of helps a little bit. And then I pulled up my spectrum viewer, and I was looking. I was like, huh, we don't usually think about it, but PI has a pretty reasonable excitation off the UV laser that PE doesn't have so much of. So when I looked at PE versus where I would expect to see PI off of the UV laser, it was really clearly still these cells and these PE ones out here were the ones we determined were alive. We didn't have very many cells to collect a bigger data file, but you can sort of see how looking at different channels, which you may not think of as an individual fluorochrome's home channel, can be advantageous as a sanity check and to verify that what you've found is the population that you think it is. Okay, so we've talked a little bit about some tissues and dissociation and approaches to those. You may end up with even weirder samples. We don't just see fresh tissues. I don't usually see FFPE, that one's pretty tough, but I do get lots of frozen. Okay. So once you get into some of these preserved tissues, things get really messy really fast. But these non-traditional samples, at least non-traditional for flow, they can be very valuable. By preserving tissues by freezing or FFPE, you can ship better to do multi-site collaborations or multiple analyses to mix together. I know we have a large biobank here at my institute, and we get samples from all over the world that the genomics core here will work with. And they get sent to like other cores too to do these large multi-center trials. So these biobanks can have really important historical, longitudinal samples. There may be rare disease or rare tissue specific banks. The problem is that cells are really disrupted by freezing or fixation and embedding. So we have to deal with things like ice crystals busting cells up, formalin fixation, paraffin embedding, and trying to do the antigen retrieval and everything from those is a high loss prospect. But there are some features that may be preserved well enough to remain useful. So as I said, like genomic assays. So we do a lot with that. I'm actually starting to get some samples that were dissociated from frozen human tissues, and we're kind of working through those, so I'll talk about that a little bit too. So when you can't really count on having a whole lot of cell integrity, but what you're after is DNA, you still have your nucleus, and you might be able to get that. So I see this predominantly with brain samples, people isolating frozen patient nuclei from frozen patient brains, like from Alzheimer's banks or Parkinson's banks. So there are a few ways you can do this, and you can do it from cells or tissue. If you just need nuclei for something like ATAC, or if you get enough nuclei, you can do some RNA-seq on them too. Right. So this is one approach you can take, is without any sucrose in it. So you would start by mechanically and enzymatically disaggregating the tissue source if necessary. And then you swell it in a hypotonic buffer and you make these puffy little cell water balloons. And you have a couple of options. You can do, with this swelling, you can do a mechanical disruption, like a dounce to break up that outer membrane and some of the organelles. And then you can pellet the nuclei. You can also use some detergent lysis to go this route to break up the outer membrane, but be very, very careful if you decide to go the detergent route, because too much detergent is going to also lyse your nucleus and break up your entire sample. So I think most of my folks actually use the hypotonic swelling, and then they dounce and pellet nuclei. If you want to do the one with a sucrose cushion, which is also what we see a lot of for brain, so this is the hypertonic, I guess it is hypertonic on this one. So this is the nuclear isolation buffer, and it does have a little bit of detergent in it. And they use a sucrose cushion to kind of catch and get a differential centrifugation. This does require ultracentrifugation, okay? But it seems to do really well on the postmortem brain tissue. So, and I've got some other groups who are starting to try these methods as well for adipose tissue. I'll let you know how that goes. No matter what you're starting with, whether it's tissues or cells or frozen stuff, always check your work. Sorting can be expensive. Um, you don't want to waste your resources and reagents if you don't have a good sample. And the best way to check your work is to look at it. I mean, I've heard a picture is worth 1,000 words. So put that sucker under a microscope. Count your cells with a hemocytometer, or if you're going to use an automated counting system, validate it first. There are lots of problems with trypan blue-based automatic counters, especially related to the quality of the trypan blue. It will not uncommonly, when it's been around for a while, start to make its own little aggregates and things that the counters count as dead cells. If you have access to a smaller cytometer that maybe is easily available in your core or in your department, I know some departments here have their own little Accuray, or they just come down and quick use my Cytoflex just to check and see, is there anything even there that's worth sorting or staining? Okay. This is a picture from 10x Genomics of what the quality of their nuclei that they would want to see. So we've got these really nice round ones. These ones are probably still okay. There you've got a little bit of fuzziness at the edges. This one's starting to look a little like, I don't know, maybe an elephant. These might be frogs, but they're totally degraded, and so these wouldn't be worth continuing with. Other times that I've forgot to pull out that microscope, if we think we've identified a target cell population, but we're not sure, for something simple like a GFP sort some into a tube, put it on a slide, take a peek. Or if the sample is running really weird, I had one that I couldn't figure out what was going on. Like it looked like it was all one population, but it wasn't running well. And I asked my user if they had taken a look at it, and they said no. So we went and did that. And it was looking weird and running oddly because it was all aggregates of about 4 to 5 cells. So singlet gates, like everything just looked weird. So we went and we looked at it and figured that one out pretty quickly. So staining nuclei. Nuclei stain a little differently than cells. In order to really help you get your data, think about a few things. So if you're doing nuclei, add a DNA binding dye if it's compatible with your downstream analysis. The only downstream assay I can think of that I would hesitate with a DNA dye about is ATAC-seq. Not that you can't make libraries from nuclei that have been stained with these, but I don't know if the binding of the DNA dye has any impact on the chromatin structure. I haven't seen that data yet. In general, 10x at least recommends not having a viability dye for that, or not having a DNA binding dye for ATAC-seq if you use that. But these are going to label nuclei regardless of whether they're live or dead of the original cell, right? Because they're all essentially dead. They don't have an intact membrane anymore. But the other reason I like it is that when you're trying to find things, having the DNA binding dye there gives you access to the cell cycle, right? So if you can find your G1, G0, and G2M kind of populations, it gives you additional confidence that you found the right population. And sometimes it gives you another way to sort of separate out cell types. So say you want the nuclei from neurons. Maybe you think that most of those aren't cycling, so they're going to be in the G1, G0 sample or population. And so then you just narrow in on that. Or maybe you've got a user who's studying cell cycle. I've got one of those, and they want to sort of see what's going on epigenetically at different phases of the cell cycle. So we can sort of tease that out using these dyes too. I've never had one of these not be helpful. Now, when it comes to staining nuclei with antibodies, looking for like transcription factors or anything like that, they behave a little bit differently and need some extra time for optimization. So they may cross through the nuclear pores without any special treatment. Nobody I know is doing a separate permeabilization step for staining their nuclei, but I see increased background for every nuclei sample I've ever seen. So here, this blue population is nuclei that have 7A-AD and were never stained with the antibody. And the pink population is a sample that has 7A-AD and the antibody. So the entire population shifts up in this new N channel. And it looks like there's a bright and a dim. And what we think is going on is that this is mostly a background. And so this bright is our positive and the dim is our negative. If you see this, first of all, don't panic, don't be surprised. It's pretty common with nuclei. But think about it, maybe do some more optimization. See if you can titrate that down a bit. A lot of nuclei staining props I see don't have a wash step because they're kind of hard to deal with. So if it's possible to add a wash step, it's worth trying. If not, again, maybe try this titration again. Ideally, you'd have a biological control, like nuclei from a sample that, like from a tissue that shouldn't have N at all. It's kind of hard to get. The other thing that I've seen people do is, like in this case, if we sorted enough, run a Western and make sure that we didn't actually detect any N protein in what we're sorting. So think about having these backup ways to validate what you're sorting and staining in nuclei. Oops. So as far as overall sample preparation considerations, I would say putting the energy into optimizing your sample prep is worth it. So think about the enzymatic conditions, which ones for how long, track those reagents so that you know and validate that your new lot or your new bottle is working the same. Think about your homogenation conditions with or without detergent. How much downsing are you going to do if you do a mechanical homogenization? Do you want to deal with the sucrose gradient or not? I'm kind of like of two minds with the sucrose gradients. They kind of mess a little bit with the sorter. Things look weird and run a little bit weird, but they do seem to help with the nuclei. So this is something we're kind of trying to work on and get a more optimized or get better knowledge about the boundaries of what sort of buffers we can use for that. If it's feasible to do a dead cell or debris cleanup or an enrichment before heading to the sorter or the analyzer, that's also helpful. I mean, try it. I actually had one where it didn't work at all, and so we're going to skip it, but it might work for you. and then it makes everything else go faster. And then you're going to want to try to keep those samples singleized so that they run best. So for cell lines, try to avoid divalent cations and use somewhere in the 1 to 5 millimolar EDTA. My standard is 2. If you have a lot of dying cells and the stringy, snot, sticky phenotype, then ignore the EDTA and do a DNase treatment. If you've got clumpy cells that you are getting out of culture after trypsin digest, try quenching with soybean trypsin inhibitor. For the nuclei, they do need those divalent cations. Keeping them cold, keeping them as stable as possible, and the sucrose gradient may or may not help. Okay, there it is. Okay. As far as instrument setup goes, I don't have to do anything nearly as complicated as Vera and Nicole. All the things that I'm looking at, even nuclei, are big enough that they show up just fine on my conventional cytometers without having to do anything tricksy. It's actually really cool on the Cytoflex. I can even see changes in nucleus size from G1, G0 to like G2M. They shift in the scatter. It's another nice sanity check. So analytical cytometry, take advantage of those DNA binding dyes. Use 2 of them if you have a messy sample to help find A, your nucleated particles, and then B, which ones are still alive. Or use them to find a cell cycle in your nuclei so you know what you're looking at. For the setup, I do like to make sure that I have really tight CVs for any DNA dye channel if I'm doing nuclei. I mean, we QC everything every day. We do have an Astrios for our sorter, so we are able to manually go align and tweak that. So I just pay extra attention during QC to whatever the DNA dye channel is. I get the best CVs with DAPI and PI, not so great CVs with 7A AD, but if that's what you have to use, that's what you have to use. So bring all the controls to make sure you can find your nuclei. Mix up your gating strategy if things look really weird, right? Go for viability versus scatter, or start with your markers and do viability and scatter cleanups later. Thresholding. So when you're setting thresholds, most of the time the defaults seem to be okay. If you're doing analytical cytometry on maybe a nucleus, nuclei preps, or cell cycle in some strangely fixed, very dirty sample, you may actually want to switch and trigger on your fluorescent dye and lift that threshold to get some of that debris out of there, the non-nucleated debris out. Okay. So that just helps you visualize your population more easily. So for sorting, we're going to do most of the same things that we talked about here for analytical cytometry would still apply. We're going to add some more considerations for buffers to help with viability, something like heapies. Pay attention to how your nuclei are running. If the flow rate's really bouncy and your stream's not super stable, then you may need to dilute the sucrose a little bit, filter again, collect them into some sort of cushiony buffer. One thing that I don't have on here is that we actually do all of our nuclei sorting on a 100. We use a 100 at 25 psi. Are they small enough for a 70? Yeah, but they tend not to come out nearly as well. So we like to go a little bit bigger. And we'll take the hit on sorting speed just to keep a larger percentage of our nuclei suitable for whatever's going on next. Sample handling before sorting. Yeah, dilute and filter, monitor. I always put up a time plot, always time versus something, scatter, time versus fluorescence, pick your favorite, whatever you can see best. So we monitor because we find that some of these samples just start to aggregate again over time and we have to refilter. If you're nowhere near the limit of where Poisson statistics starts killing sort efficiencies, sometimes running a little bit faster can be helpful. So if your EPS is low enough, kick it up a little bit. and see what happens. Sometimes the shear forces of it entering the instrument can kind of disrupt some of those aggregates a little, and it'll smooth things out and get your efficiencies up. Now for sorting, I'm going to say the opposite kind of what I said for analytical cytometry about thresholding. Thresholds are great, but be very careful changing thresholds when you sort, because anything the cytometer can't see or ignores, it's not taking into account when it's making a decision of whether or not to attempt to sort the drop or to abort that drop. Okay, so anything that you've threshold out and your cytometer can't see can end up in your tube. So if you're thresholding out stuff that isn't going to negatively impact your downstream assay, cool. Speed up your sort, make your life a little easier. But if the things that you are thresholding out will have an impact downstream or could interfere with something, then you're going to have to change and keep that threshold much lower and just deal with having the extra noise in your data. This did not take me as long as it did yesterday. So I feel much more comfortable having conversations and question stuff anyway. So I think I will just go to the next slide, which is just thank you, MetroFlow, for inviting me to talk, and ISAC for support, UW Flow, because that's where I got started in Flow, and they taught me a whole lot there, and I love it, and the Van Andel Institute for letting me play around with their cytometers. I know I've seen a bunch of notifications pop up, so what would people like to know about? What sort of questions do we have?
Okay, thank you so much, Rachel. Awesome. That was definitely really eye-opening about a lot of different tissue types and all of that. And I think we do have a couple of questions that came through, so this is a perfect opportunity. And thank you for an awesome talk. So our first question that came through from Alessandro says, thank you, Rachel. And if you can please let us know, what is the more adequate collagenase type for improved HSC characterization after a bone marrow flush?
I don't think ours are doing any collagenase. I think they just flush and filter for all of our bone marrow samples.
Okay, so then from there, we have a question from Ming that said, nice talk, Rachel. And what is your favorite permeant DNA binding dye? And would it affect any downstream analysis?
My favorite cell permeable one. Well, that depends on your instrument configuration and what else you're running. I mean, I am a fan of Dicycle Ruby. I like it. It seems to play really well. It's worked in the panels that I've needed to use it for. And the only thing I would be concerned with for downstream is ataxic or anything that's going to look at chromatin structure. So this gets really complicated, and we have discussions with users because a lot, because so much of my user base sequences. That we do a lot of flow upstream of genomics core because we have a phenomenal genomics core here. Most of the time, having something like a viability dye, a DNA binding dye in your sample is going to have no impact on sequencing for a number of reasons. If you're using an impermeable dye, like DAPI, PI, 7A-AD, only your dead cells have it in any appreciable quantity. And then your live cells that get sorted out have, like, what's in the solution is only whatever diluted from the size of the cell buffer drop surrounded by the sheath drop. So it's getting diluted out. And then you wash them and you dilute that out. And even if you had something that had a DNA binding dye to it that was permeable, as soon as you go through that process and you start diluting it, it's a reversible kinetic process. So those dyes do start coming out during processing. So as I said, the only thing I can think of off the top of my head where I would be concerned would be ataxic. And I still think 10x recommends against having it in your nuclei. Again, for live cells, you need to know what's alive.
Great. We also have a question that came through from Wei, and they're asking, how do you balance the sorting speed for 96-well plate sorting for nuclei? Essentially, single cell sorting for nuclei. What speed would you recommend?
What's the frequency of your target population? Honestly, the Astros does pretty well with plates, and I have yet to find, like if it's a reasonable sized population, then I just kind of run it until we kind of max out how fast the sorter will go, which doesn't take long. I think my Astros takes 54 seconds to sort a 96 well plate. So
Yeah.
I don't.
Oh, sorry, there's a follow-up, sorry to interrupt. They said that they have 0.5% positivity for Ruby to answer that follow-up that you had.
0.5% positive?
Yeah.
I don't know. I kind of base it off the nozzle. So with a 100, I wouldn't go over 5,000 or 6,000 per second. But again, your mileage may vary. Watch your sort efficiencies. And if you notice that your sorter is, it's moving the plate as fast as it can, then you can slow down.
Yeah, that makes perfect sense.
Mm-hmm.
Okay, so then we have a question from Claudia that came through that's asking, if you have any experience with this, what digestion enzyme do you recommend for murine skin? So skin preps.
Ooh, I don't
Interesting
know.
one.
I have not actually had anyone do that. We do, a lot of my users use our Miltenny Gentlemax. Gentlemax? Yes. The Gentlemax Octo with heaters, which is really convenient. It's kind of like a mini blender that has a coffee warmer around each tube so it can process samples in parallel and you don't have to go in and out of the incubator. So Miltenny has a lot of different optimized protocols and cocktail kits, so it may be worth seeing if they have something for skin to see what enzymes are in there. And then again, Worthington Tissue, the Worthington Tissue database handbook thing.
Yeah, that's definitely a nice, big, good resource.
It is. They have so many different tissue types.
Yeah, I used to love having that at my desk. So we also have a question coming through from Tina. Do you know how your users in genomics are evaluating post-sort count and viability prior to processing the sorted cells on the 10x system?
I do. We have a collaboration. So I will sort things and then the user will go off and count and all of that and then bring it back to genomics, and they require some minimum volume, and I get 50 microliters of it, and I add 50 microliters of a DAPI-based flow buffer, and I run it on my Cytoflex, and we look at, with a fixed threshold, by the way. The Cytoflex by default has this weird wandering threshold, which is not super helpful for this particular application. So we run it on there and we report back to the genomics core approximate cells or nuclei per microliter, percentage of those that look dead, and then how much debris and noise we saw. And we realized that we're going to have noise and debris kind of overlapping. So we're just kind of hoping to track trends on that one. But it's more important that the count is good and the viability is pretty reasonable.
That's actually really nice, because I know a lot of genomics cores have their own separate, and sometimes it can be a little bit different across the cores. So it's nice to hear that you have that collaboration. That's awesome.
We're literally across the hall from each other.
Yeah, that's great. Again, another circumstance where I'm slightly jealous.
I'm more jealous of like going to sea and sorting on a boat.
Yeah, that's what I'm saying, for both circumstances. So then Evan has a question. For nuclei from frozen sections, do you find that you need to up the nozzle size because they're extra sticky or fragile? And it seems to help them up in Connecticut for with tiny nuclei but larger nozzles because of those frozen sections.
Yep. Ours are coming from frozen sections. They are delicate. They are still sticky. These are from brain because brain is like the worst. So we do. That's why we run on the 100. Even if they're technically probably small enough to go on that 70, we didn't get good results.
Yeah.
So the 100 worked a lot better. I would like to try them when I get my new sorter soon. To try them on a 130. But right now, all I have is the Astrios, so my next size up is a 150, and it's miserable
Yeah.
to run, so I haven't done that yet.
Yes. Been there, tried that, and stuck
Yeah.
with 100.
Yep.
Claudia just wants just a little bit of clarity. She wants to just make sure if you mentioned about DNase treatment and that it can't be combined with EDTA. She just wanted to make sure.
Nope. If you're doing DNase, you cannot add EDTA. DNase requires magnesium to work.
And then I have one or two questions. So I was curious if you've done, any work with any type of neuronal preps from mouse brain or anything like that, and sorting them for culture as opposed to sequencing. And if you've noticed poor viability post-sort that might require sorting onto a layer of other types of neurons like astrocytes or anything like that. It's something that I've done in the past that it's always been very tricky, and I don't think we ever like fully optimized. It seemed to work out in the end, but curious about that, like sorting onto other tissue types for neurons to grow.
So far, everyone's been doing sequencing from the neurons that I've been sorting, or the astrocytes, depending on what lab. I would believe that they are finicky, and that you would have poor post-sort viability, because you've had to take them out. They're losing all their little processes. It's a rough ride for them. So I like the idea of having a feeder layer if that's feasible. If that's not feasible, my recommendation for cells that are hard to culture post-sort, especially like for single cell stuff, is conditioned media.
Yeah.
Lots of cells don't like to be lonely.
Yeah.
So you can give them conditioned media, and sometimes it tricks them into thinking there are friends around, and they start to grow.
Yeah, I love that way to put it. Yes, absolutely. And then another question. So out of curiosity, there are, all the different types of tubes that people can, or there are different types of tubes that people can bring. Do you have a preference between polypropylene and polystyrene for final resuspension of cells and also for collection tubes?
We tend to use polypropylene because we hate chasing down multiple tube types, and it can be easier for cells. Again, if you think that the plastic type is going to be a big problem, I would try it head to head and see what happens. For plates, they're almost all polystyrene that people bring us. The only thing that I'm really, really strict on, though, is any collection tubes you bring me need to have something in them. Dry plastic and static charged little tiny water drops don't get along very well, and we can watch them like jump back out of the tubes. So just rinse them down with something. Make sure you have some buffer in there. So, if you're going to do culture, 100% FBS tends to be what we put in the tube, because it's going to get diluted out as we put cells in the tube.
Okay. And then one more question. So, you talked about the use of like a viability dye and also some type of vital marker to pull out live cells from debris as opposed to dead cells. Do you ever kind of add to that, any type of apoptosis marker as well, and like kind of hit it with everything you've got if you, or not really?
Haven't yet, but that's on the list. We've got some of my colleagues here have developed a really cool single cell sequencing method, and we kind of want to add in a nexin to eliminate the apoptotic cells, because the cleaner we can get those sorted cells, the better it's going to be for sequencing, especially for RNA-seq.
Awesome. Perfect. Well, I think that leaves us right at about 12:40, so that's perfect for questions and timing. And thank you again so much, Rachel. That was again a wonderful talk, and we really appreciate you being here today.
Thanks for having me.